Hi, I'm Bill Chin. I'm an internist and endocrinologist, a longtime faculty member at Harvard Medical School. I founded the genetics division in the Department of Medicine at the Brigham and Women's Hospital, was an investigator in the Howard Hughes Medical Institute, and an employee of Eli Lilly and Company, where I ultimately led global research. I'm currently the executive vice president for clinical and translational science at a startup company called Frequency Therapeutics, and the Bertarelli Professor of Translational Medical Science and Medicine Emeritus at Harvard. So what is a biomarker? It is a characteristic that is objectively measured and evaluated as an indicator of normal physiologic or abnormal or pathologic processes or pharmacologic response to a therapy. There are many types, and I don't have time to go through all of them. The first one is diagnostic, which in a way speaks for itself. Another is predictive. This is before you have a diagnosed disease and could reflect a risk or susceptibility for disease. Another would be prognostic. So after you've had a diagnosis given, to understand what is the natural course of your disease or how you might respond to a particular therapy. In addition, there are safety markers and also markers that indicate where and when a drug will be effective in the blood. So biomarkers are very important in drug discovery and development. They are incredibly useful in decision making. For instance, if you talk about animal studies, how do you do the right animal studies? How do you choose the candidate molecule for further development? And finally, how do you know when to have an effective and efficient trial in the clinic? Now, one example from my own career focused on something known as personalized or precision medicine. And the area of interest was cancer therapies. So the issue is, how do you get the right medicine in the right patient at the right dose with the right schedule using genetic and biochemical markers? The disease of interest here is non-small cell lung carcinoma, which is found, unfortunately, in patients who smoke and also non-smokers. And there is a mutation in one of the genes, a somatic mutation. The gene is called the epidermal growth factor receptor kinase, and mutations in these genes often can lead to this cancer. So many groups have found medicines for this mutation, and so if you can match that drug with the subset of patients with this kind of cancer, then you really have achieved personalized precision medicine. The other example deals with hemoglobin A1C. This biomarker is very important for the diagnosis and treatment of diabetes. We now know, after nearly 50 years of experience, that the hemoglobin A is glycosylated, or a sugar moiety is added to the N-terminal amino acid, or valine, of one of its chains, beta globin. This effect can reflect plasma glucose levels that are integrated over time, which gives you a sense of sugar control over a period of as long as 3 months, which is the lifetime of this hemoglobin A. In sum, biomarkers are critical for efficient, effective drug discovery and development. The trick really is to find reliable markers to help you know whether you're on the right track. So this would save time and money, and most importantly, be able to bring medicines to patients sooner.